So factors that influence enzyme activity are the things you might expect: the pH, or acid versus basic level of the water solution in which the enzyme works, generally inside of a cell, but not always. Enzymes can be sent out of the cell to do work as well, like in our stomachs, or when a fly eats, it regurgitates enzymes onto its food and then sucks up the food like a straw. So pH, very important. Temperature, I already talked about temperature, but temperature is really important with enzymes. Too hot, you'll destroy the enzymes. Too cold, the enzyme activity falls below some functional threshold. It still might work, but really slowly. And at some temperature, it stops working altogether. And the presence of ions or minerals. Now, we're not talking about the coenzyme or cofactor when I say ions or minerals, other ions or minerals in the environment. Excuse me. So enzymes are easily inactivated, easily turned off, or denatured. And the word denatured means that we're doing something that messes with the three-dimensional structure of the enzyme. Now, the original protein that makes up an enzyme is a string of amino acids, but they're folded in a very precise way. You take a piece of paper, for example, and it's just a piece of paper, but you fold it just the right way and you have a hat or a swan or any number of things that an origami artist can create out of a piece of paper. And proteins are the same way. They're folded in a very precise way to give them their function. And if you change their shape by changing the pH, by changing the temperature, by changing the ions or minerals in the water solution with that protein or enzyme, it will lose its folding. It will possibly unfold completely, or possibly just change enough of the folding where it's no longer functional as it once was. And we call that denatured. I think the most familiar protein denaturing that you experience through life is cooking eggs. What is an egg white? It's not white at first, it's clear, right? It's virtually clear. And it's almost 100% protein. But you apply a little heat, either by boiling a hard-boiled egg or frying the egg in a pan, and what was clear is now more solid and white. What happened? The proteins in the egg white were denatured and changed their form and function, and they never go back. You can't redo it. Unlike a piece of paper, if I fold it into a hat and I unfold it a little bit, I can always fold it back. You can't do that with protein chains. Once you've denatured it, it's no longer functional. And so we can denature enzymes using or manipulating pH, temperature, or some of the chemicals, the ions and minerals around in the solution. There are other types of enzymes, one called protease, which is a very generic protein enzyme. And what does it do? It breaks up other proteins. It just eats them for lunch. So protease is a general digestive enzyme or catabolic enzyme that breaks down proteins, and it will destroy other enzymes as well. But there are specific inhibitors. If I'm trying to stop protease, I want protease in my sauce that I'm making. I want protease activity to stop. I don't want it to continue to break down all of my other proteins. So what do? I add an inhibitor. I add a known chemical, a known substance, which will interfere with or denature the protease. And there are also amylase and other types of inhibitors that you can use in your processing to stop the enzyme activity from continuing. It's not just cooking. So enzymes usually end with the ASE suffix, protease, amylase, etc. They are usually named after the substrates they act upon. So example, urea, which is a protein compound in everybody's body. It is a product made from the breakdown of proteins. It's also a great fertilizer. You can fertilize crops with this. But if you take urea and put it in the presence of an enzyme that breaks it down, that digests it, you call it urease. Again, the substrate plus ACE is commonly the name. There are lots of exceptions, though. So lactose is broken down by the enzyme lactase. Lactose is milk sugar. Now, there are the resulting type of chemical reactions, so not the substrates, but there are a number of chemical compounds that can be hydrolyzed, or the chemical process is called hydrolysis. So these general hydrolysis reactions are pushed forward by hydrolases. And you see hydrolases because it's a category. There are lots of hydrolases because there are lots of different substrates that can be hydrolyzed. Oxidation, oxidases, the same concept. There are lots of different oxidases or oxidating enzymes. The rule doesn't always apply. For example, phycin is found in figs. Papain is found in papayas. So their name is from the origin and they don't end in ace. Well, historically, some of the naming conventions hadn't been established, or certain research regions around the world weren't following the conventions the same as they were in other countries. So we have some very important food-related enzymes, phycin and papain, that are specific proteases. Excuse me. Properties of enzymes. What do? Well, for the plants, they're controlling ripening. They're doing important work. Starches are broken down into sugars. Pectins, which are substances that are in high levels in apples that are unripe, the pectin goes down as apples ripen in other fruits. But over time, food will spoil. There's too much of this. And think of it from a natural standpoint. You have a tomato, what is best for the tomato plant? With no humans around, to extract the seeds. You want that tomato to ripen. Maybe some tomatoes are eaten and the seeds spread through the poop of other animals. But what if that tomato drops on the ground? You want the ripening process to keep breaking it down and disintegrate the fruit, leaving the seeds in a rich environment of nutrients where the seeds can start to grow. So it's an evolutionary necessity in most cases for fruit to ripen and then get overripe and then allow this next generation of seeds to grow. Now, in this case, with rotting, they're talking about enzymes that are being excreted by the cells, the little string-like cells called hyphae from fungi. So if you've got mold on your food, how is that breaking it down? Enzymes from the other organism, the mold organism, is breaking down your lettuce or your broccoli or whatever. Enzymes are responsible for changes in flavor, color, texture, and nutritional properties. In many cases, for the better. You need the enzymes to do their thing in order to get that right, correct flavor, color, texture, and nutritional qualities, releasing the nutrients from the matrix of the tissue that you're ripening. But at some point, you hit that sweet spot. Maybe literally, maybe figuratively, but you reach that maturation where it is where you want it for the consumer. And then you want it to stop. Enzymes can be inactivated by heat to extend storage stability of foods. We've already talked about that at length. Another way to inactivate enzymes is cooking. Now, cooking can enhance many food products. It can destroy certain properties, like destroying the enzymes in spinach leaves with heat is going to destroy the properties, the texture, and the appearance of the spinach leaves where it's no longer suitable for salad. It's now limp and a very different product. Enzymes are used for fermentation. So yeast feeds on starch or sugars or other types of materials in the food, and it releases, and it releases what? One of two things usually, an acid or an alcohol. If I'm making acid, maybe I'm making vinegar. Yeast makes vinegar. If I'm releasing an alcohol, maybe I'm making wine or beer or some other fermented type of alcohol, a hard alcohol. Hard alcohol, you first ferment and then you distill. You concentrate the alcohol. Enzymes can be immobilized to a surface of a membrane or other inert object in contact with the food being processed. Just like I described earlier with the catalytic converter. You can put these catalysts on metal plates in your exhaust pipe to help chemical reactions occur so the unburned fuel that makes it through the engine into your tailpipe can burn on the catalytic converter before it actually goes out the end of your tailpipe, making it cleaner. Well, how do we do that? We created metal plates coated with catalyst. We can use metal plates or plastic plates or other kinds of membranes and coat them with enzymes. So these membranes are coated with enzymes that now can be used in the food processing. So we're running some sort of food grade material across these membranes, through these membranes, however they're made. And the catalysts are there to speed up the reaction that we're trying to have occur in our process. Lots and lots and lots of enzymes are used in food processing. Sometimes they're just added as a powder, and sometimes they are immobilized on a surface so they can keep working as product moves through the system. Enzymes can be extracted and purified to a high degree if needed, and there's a whole commercial industry on that. You take raw materials and you extract and purify enzymes and turn around and sell those enzymes to somebody else. So if I'm making a fermented product, I have to buy the packets of yeast or packets of enzyme material that I'm adding into my recipe. I got to buy them from somewhere. So there's a whole industry on extracting and purifying food grade enzymes. So please don't try to memorize this. I don't care about this list in terms of testing, but look at them in terms of understanding, recognizing a couple of the words, but this goes beyond what I need for this class. Understand that there are lots and lots of different reactions. Industrially, most are protein hydrolyzing enzymes. Nearly 60% of the industrial food reactions are protein hydrolyzing reactions. Carbohydrate hydrolyzing reactions, lipid hydrolyzing reactions, and specialty reactions in other areas, analytical like research, pharmacy. And some numbers that we don't need to go into. These are older numbers anyway, but I'm not going to take the PowerPoint slide out. You can get an idea of some of the important industries. Tons of money spent in the bakery industry. Tons of money in the beverage, dairy, fats and oils, culinary, meaning cooking, preparing food, restaurants, and meat industries. The US market has a massive market for enzymes. Plus some of the competitors. Don't worry about memorizing competitor names, not a big deal. Annual sales, again, just some numbers to get an idea. Things you might not have thought of. Detergents, you're doing laundry. One of the things that can be done is adding enzymes to help break down certain things that would be stains on your clothes. So using enzymes to improve detergent activity, or enzymes in the manufacture of the detergent itself. So the enzyme might be actually helping the detergent work, or it might be helping to make a detergent. High fructose corn syrup, largely in the candy and soda market, especially sodas, billion-dollar industry. Important factors in using enzymes. There might be reactions you're trying to have occur. You want to create a reaction that isn't realistic under lab conditions. You're lowering the temperature, making something happen that you normally wouldn't happen without that enzyme. Allows milder processes. So you can do it at lower temperatures, milder pHs. You can reduce the number of process steps required to get your final product if you include enzymes in some cases. This is really important in pharmaceuticals. reducing organic solvent use, which organic solvents have their own problems. And if you don't have to use organic solvents, if you can use enzymes instead, it is a safer, more predictable product. Genetic engineering has been done to improve enzymes, make them even faster, lower activation energy, whatever. A lot of genetic engineering has been used to manipulate enzymes to work more effectively, or manipulate yeasts. Bread making, flavor development with lipases. Fats or lipids, lipases break down lipids. Fats are huge in our sensory perception of flavor. And so lipases change the flavor profile by changing the lipid profile in the food product. So you can really change flavor with lipases. Cheese making, clarifying fruit juices so that they're more clear. You're breaking down substances that would make the juice cloudy. Textiles or fabric and carpet and other types of cloths and threads are made with the use of enzymes. That's stone-washed jeans. That's an enzymatic process. Grain processing, all kinds of stuff. Waste management, cleaning drains with lipases. What is clogging up a lot of drains? It's usually a buildup of lipids, of fats. And so lipases included in drain cleaning can be important in waste management at the consumer and industrial levels. Even the health food market, food enzymes that are sold to promote digestion. Do you need these? Probably not. If your doctor doesn't say you have a specific enzyme issue, you probably are wasting your money, but there's a market there. Again, please read through this, making of syrups, making of fermentable products like brewing or distilling alcohols, beers, malting, malt flavor. You ever had malted milk balls or malted milkshake or something? The malt product is made with a fermentation type process. Meat tenderizers, things like bromelain, papain, and physin. Remember those words from earlier? Papain and physin. They're important ingredients in some of the meat tenderizers or some of the seasonings that you might get at the store that are for making marinades for that steak, and these beautiful meat spice mixes. Well, some of them, many of them contain papain and or physin in order to not only add flavor with the other components, but to help break down the proteins a little bit as you're marinating it so that it's a little more tender when you're done cooking it. Lipases, flavor production in chocolate and cheese. So have to mention that because it's got chocolate and cheese. Glucose oxidases, desugaring of eggs, flour, and potatoes to prevent browning. So you're removing the components that will turn brown, making an ugly product, and browning also can change the flavor. And so by desugaring, by using glucose oxidase, it is a more stable product that won't turn brown. Very common also in use of salad dressings. Pectinases. Pectins come from, like apple juice is a great example, very high pectin in apples, and pectin will make the juice cloudy. It also can help increase the yield of juices by breaking down the pectin. You're releasing more juices. You're helping to macerate the cells better, to break things down, to get the juice out. So the use of pectinases helps you increase your overall juice yield. Bleaching of flowers, quality testing. Again, just take a look at these. Flavor aces, very generic category for improving the flavors. Anything else? This is just a cornstarch processing diagram showing how cornstarch is used and some of the steps to make this high fructose corn syrup. And again, I'm not going to go through some of this. Just hit pause or go through the original PowerPoint. Inhibition of enzymes, some things that you should know. Sulfite, I've talked about that before, but it stops certain reactions, especially oxidative reactions. And so sulfite is used to keep white wines clear. It's used to keep the nice orange color of dried apricots and other fruits that are dried. pH, such as vinegar and citric acids, can be used to inhibit enzymes. EDTA is a preservative that binds to the copper, making the mineral copper unavailable to function properly with the enzyme. Vacuum packing, you're removing oxygen. So without oxygen present, that can inhibit enzymes that require oxygen to complete the reaction, but not always. Depending on the product, there might be enough oxygen in the cells already to do damage. So vacuum packing does minimal good. It helps, but not enough. So sometimes you need more than just vacuum packing, but these are just some examples. We talked about blanching before. That's a quick heat dip from a few seconds to a couple minutes, depending on the product. But you're not cooking it. You're just exposing it for a very short duration for surface cleaning, surface reduction of the bacterial and fungi spores that might be on the surface, and deactivating of enzymes in the cells, especially near the surface. And irradiation also can inhibit enzymes. Again, I'm not that concerned about these slides. These are really important slides for people who are actually going into the culinary or food manufacturing industries, because you want to know about these enzymes. You want to become an expert in them. For the level I expect in this class, there's just a lot of examples, and you should be able to come up with some of these examples. You should be able to know some, but I'm not going to ask you about all of them. Don't memorize this list at all. Just know some actual real uses of real enzymes, how they can be used in foods, and know a couple of enzyme names. Certainly the ones that I spent time on in the earlier slides, I should say, you should know more of those. In the later slides, when I started skipping through them pretty quickly, just look at them. If you're interested, go a little deeper, but if not, you should just be roughly familiar with the kinds of uses that enzymes typically have in industry. And just to make one last reminder, it's not just the food industry. And I almost guarantee you, whatever device you're using to watch this video, there were enzymes used in the manufacture of something in that device. Something you're wearing was probably manufactured with enzymes somewhere in the process of what you're wearing. Your car, everything imaginable, it seems there's enzymes somewhere in the process, either that are naturally occurring as part of the process or that we intentionally add to alter, aid, or speed up the manufacturing process.